All right, we are going to start this benchmark on DNA replication. This is the first part of our unit on cellular division. So the first thing we're going to point out is about replication. All right, we should know that to replicate means to make a copy. So in this case, what we're copying is the DNA. So DNA replication is the process of making a copy of DNA. And obviously, this is going to be necessary for proper cell division, because if what you have is 46 strands of DNA, when that cell divides, what you want are two cells with 46 strands of DNA. That extra strand of DNA isn't just appearing, or those extra strands, 46 extra strands, aren't just appearing out of nowhere. They're going to be made through the process of DNA replication. So, what's going to happen is that one side of the DNA will start to serve as the template. The blue strand is going to be built based off of the red strand. So it depends on what the strand says. So if you look here, that side says A, so the blue side knows that it needs a T. Versus here, an A is on this side, so the blue strand needs a T there. All right, this one is equal to this one. All right, so you can see AT, AT. All right, as our cell, or as our DNA is starting to replicate. The first thing that's going to happen is that the DNA helicase is going to unzip the strand of DNA. So if we go back and we look, we could say that right here we have DNA helicase. That DNA helicase is moving in this direction, and as it goes, it's pulling apart the DNA strand, or unzipping it. It's kind of like when you have a zipper and it tears apart in the middle. That's kind of what's happening here, all right? Our strand of DNA is going to open up. So what happens is that that strand of DNA, forgive my bad drawing, is going to open up. We're going to have a big giant opening. We've got a DNA helicase. We've got a DNA and a helicase. We can have multiples. They're going to move in opposite directions and really just start to open up the DNA strand. Each of these 2 points, right, is going to be referred to as being a replication fork. All right, a replication fork, a fork meaning like a fork in the road where it splits. If you look here, this is where it splits. This is where it splits. So it's the area where the double helix is starting to separate. All right, our helicase, all right, helicase is going to be ACE, should tell us that it's an enzyme, right? And we should know that all enzymes are proteins. And we should also know that proteins are made of amino acids. This should all be review, hopefully. Step 2 is that there's going to be a new enzyme, all right? This one is going to be called DNA polymerase, sorry. Remember, a polymer. Polymer. We had that in the biochem unit as well. Polymer meaning many subunits, all right? A monomer being a single subunit, a polymer being many subunits joining together. So in this case, we have DNA polymerase. All right, that DNA polymerase is going to have an important job of it's going to add new nucleotides to the open strand. So basically, it's going to start to form the new half to the old half. So what we're going to have, again, my bad drawing, we've got our strand of DNA. We have our helicase. Only now, we also have polymerase. And there's going to be multiple polymerases as well. Polymerase is going to be following right behind helicase, adding the new half of DNA. Adding the new half of DNA. And you can see we're slowly starting to assemble. We still have the other halves, right? Here's our DNA, all of our bases. that are joined together by hydrogen bonds. We should know this stuff. So the helicase breaks those hydrogen bonds, and polymerase forms new hydrogen bonds with new nucleotides. So we can draw some of the free-floating nucleotides, those free-floating pieces that polymerase is going to have to put into place and attach to build our other half of our strand. This is going to keep going until what we ultimately end with are two separate strands of DNA, and the two separate strands of DNA are going to be half old versus half new. Right? They should also be identical. And the reason that they should be identical is because if we think about what our strand was from the very, very beginning, all right, we can think of our strand and we can say, okay, well, if A was here, then T was on this side. If C was here, G, remember, our straight letters go with straight letters, curved letters go with curved letters, adenine with thymine, thymine with adenine, cytosine and guanine go together. So we can start to look at our strand. As our strand forked, right, because of our helicase, we started to break apart those hydrogen bonds. And what we wound up doing is that we started adding new nucleotides to the other side. So that when all was said and done, what we should have had would be two strands that were absolutely identical, but half old, half new. See how they're identical? And it's also important for you guys to understand how errors can occur. DNA polymerase is able to proofread its own work, right? But just like when you proofread your work, sometimes you miss a mistake. But what happens is that DNA polymerase has the ability that they can only add new nucleotides when the previous one is correct. So the first thing they do is they look back at their old work and say, does A go with T? Yep, okay, that's correct. Moving on, all right? If it's wrong, polymerase is going to backtrack. Looks back and says, A goes with A. Oh, wait, no, that's wrong. Polymerase is going to backtrack, is going to pull out the incorrect nucleotide, and it's going to replace it with the correct one. All right? This proofreading mechanism that DNA polymerase has eliminates most errors. But like I said, all right, even when you proofread your own work, you're not perfect. So polymerase is going to, every once in a while, miss a mistake. Other thing to think about is how quickly DNA replication takes place. All right, DNA replication is going to constantly be adding new nucleotides, all right, and that new nucleotides is going to wind up being the new half of the DNA strand. But those multiple replication forks, all right, multiple replication forks, so like about 100 points, is going to be what's responsible for speeding up. So meaning, if we have our DNA strands, we have an opening, and we have another opening, and we keep having other openings in various places, that means that those openings, all right, our helicases are going to meet one another. And when they meet one another, the whole strand will open up. So it can happen a lot faster. It doesn't have to work all the way down one strand. That wraps it up for our DNA replication.